the director of academic innovation at the Riviera Ridge School. And what that means is that I work with pre-K through 8th grade teachers in STEM integration. So science, technology, engineering, and mathematics in their classroom, and really helping them come up with innovative ways to make science and mathematics interesting for students. I did this summer research experience because it was so outside of my normal skill set. And I'll talk a little bit about that in just a second. And I had a great advisor, which is Audra Di Stefano, and she's a doctoral candidate in the Department of Engineering. So at Riviera Ridge, we really focus on not only STEM, but also the arts, and we call that STEAM, as well as Kaleidoscope, which is our program on teaching religious thought, multiple religions, and all their views, and then social emotional learning. And we focus on those three areas because we believe in empowering individual potential and inspiring purpose beyond self and cultivating social responsibility. And one of the exciting things for me about coming to the UCSB program was right as I met Audra, I was asking her, like, why did you get involved in this type of work? And she said, because it was hard. And I just really connected to that because that's kind of why I did this program as well. Scientifically, I'm trained as an ecologist, specifically a landscape ecologist. So I work at a 55-kilometer scale, which is very big. And Audra works at a nanometer and microliter scale, which is very small. And so this idea of something being hard and stretching ourselves beyond our potential, what we think is our potential, is kind of a personal ethos of mine. And it's also something we practice at my school and teach students to do. So one of the things I had to pop into right away is to understand what a polymer is. And just basically, a polymer is many units. So everything is made up of polymers, and they're repeating units of monomers, which is a single unit. We were working this summer with block polymers, meaning that we had these repeating functional groups next to each other as we were synthesizing or building out our polymers. All polymers can self-assemble and self-organize. And that self-assembly generally depends on their size, how big they are, their geometry, the shapes that they are, and the external environment that they're in, especially when they're in certain solutions. Self-assembly can be really diverse. And this is a area that I think it's only maybe like 10 or 20 years old where people have really started studying self-assembly. But these diverse shapes can be made because of their ionic micellar structure, meaning that they have charged head groups. They can be hydrophobic and repel away from water. So they're kind of creating these little cells or spheres. They can also create sheets and other shapes as well. Polymers can be natural or synthetic, and some synthetic polymers are bio-inspired. And Audra's group is the Siegelman group, and they are often, many of them are working on bio-inspired polymers. So the one we were working on is a polypeptoid. And a polypeptoid is based off of a polypeptide, but instead it has an N-substituted glycine backbone. And these peptides are built on carbon, and peptoids are built off of nitrogen. I kept trying to wrap my head around, well, what is the major implication of this work? We're doing all this small work that I can't see, and inside these little tubes, and they're clear, and there's something in there inside this solution, but I can't see it. I don't know what it is. And I started looking at what's the big deal about self-assembly. And the general big deal is that through evolution, it's taken billions of years for polymers and molecules to self-assemble and create new objects, new things, right? New living things. And if we can engineer that, we can do what we've done with evolution in a shorter time span. So it's taking something of a long scale and making it in a quick time scale. So for me, that's fascinating. So the first study we worked on was the water dynamics around charged peptoids. And in this study, we tried to measure how water moves around something called a spin label. And I'll go into that in just a minute. So first, we synthesized the peptoids using a robotic synthesizer called the Prelude. And we added this spin label. And a spin label is an organic molecule which possesses an unpaired electron. So water could possibly move around it differently. We completed a titration on the peptoid one to get the peptoid in different pH solutions, and we took samples. We observed the water dynamics around the spin labels of the polypeptoids using spectroscopy, which is the ODNP machine. And atoms and molecules generally have a unique spectra. Therefore, measurements of these spectra can be used to detect their behavior. So this is what we built for this one. And you can see the spin label here. And after we synthesized it, we used something called MALDI. And MALDI is an analysis of the peptoid sample to identify its molecular mass. So we knew what its target mass should be. And then we were looking for that similar target mass after we did the MALDI. If it was somewhere close, we knew that we were in the general ballpark. And those peaks represent what the MALDI is seeing. So there might be other parts of the product that didn't join, or like kind of other things that might be in your sample. And we had promising results from this pH titration. So basically, as we added NaOH, the solution became more and more, you can see it moving up the scale here. And then when we looked at the water dynamics of those different samples, so each one of these bars represents one of these stars, you can see that the water went faster over time. And then we had some kind of plateau, and you can see a dip here, and there more work needs to be done here. So the thought was that this would kind of even out, and it might, just depending on our sample and our scale, we kind of need to go back and do it a second time. So in study 2, we aimed to measure the self-assembly of 2 synthetic polypeptoids by documenting their micelle size. And I'll show you a picture of micelles in just a minute. And we were looking specifically at their diameter as well as their size distributions. Secondly, we hope to control that ionic micellar structure using a monomer sequence. And I'll show you our sequencing. Our work contributed to the body of work on self-assembly by documenting the size of distribution of polypeptoids, NDC, NCE, and NME. So here we synthesized 2 block peptoids using repeating functional units. So these are our main functional units, and then this is the spin label that we put in for one of our peptoids. So the very first peptoid that we created had this order of NDC, NME, NCE, and then some more NMEs. And it's a very long chain. And it was really interesting for me because when we did this on the Prelude machine, sometimes it took like 36 or 40 hours to create these because they're such long chains. So you're programming all of this into the robotic synthesizer, and it's doing it one at a time for you. So you're not sitting there in the lab coming back and rinsing it or adding the next monomer. It's kind of adding it over time. And I like to think of it, because I'm an ecologist, of like, when we look at coral's kind of free floating in the environment, but then it will find a substrate, like a piece of something, and it will drop onto it. One piece will, and then another piece will, and then another piece will. So we were making that happen with the synthesizer. And so it stays in this chain. So this guy, NCA, is not going to move over here, or this guy's not going to move over here. They're all connected like a caterpillar. The other one we made was this, and this is where we put the spin label in. And that's going to help us with a water dynamic study later on. So polymers go in chains, but then because they self-assemble, they often get tangled up with each other as well. And when they're in solution, they can make something called micelles. And generally when we draw those out in 2D, we draw them out in this really clean looking way, this understanding of like kind of, it has a core, hydrophobic core, so it's kind of afraid of water here. And then as we go out, we call this area the corona, and they look beautiful in this picture. And in my mind, I was trying to visualize these because it was something I couldn't see. And I realized that this charged head group at the end was actually allowing the corona to extend when we place the charged head group charge in the closer to the hydrophobic core. This corona was not able to extend as much. And so we can do measurements to see the difference in diameter of this micelle compared to this micelle based on these charged groups. The other thing I tried to do, sorry, was visualize this in 3D because I was having a hard time wrapping my head around what exactly we were seeing or building because I couldn't see it. And so when I started working with it in 3D, it started to make more sense of these things are kind of like giant balls of spaghetti. They're not these clear cut cartoons, right? And our world is actually in 3D as well. And so that really helped me understand what we were kind of synthesizing and creating. So we prepared our amines in this thing called a prelude. And the prelude is this robotic synthesizer. And we started out with these organic compounds and they're derived from ammonia. And NCA came from salt form. So we used a free base to remove that salt and get them into this usable form. And then we programmed the robotic synthesizer, this prelude, for making peptoids by adding our sequences to a resin ball. We removed the polymer from the resin ball. And then we had to analyze our work to make sure that we had the product that we thought we did, just like the first study. And then we dissolved the peptoid into water and adjusted the pH to 9 using NaOH, where the critical micelle creation was reported. Next, we used dynamic light scattering to determine the diameter of those micelles that I showed you that we kind of visualized and figure out if we made them or not. And molecules in suspension cause light to be scattered at different intensities. And analyzing these variations, you can determine the Brownian motion of the molecules. Then the size is determined using the Stokes-Einstein relationship. But what was interesting about working with this dynamic light scattering machine is that you are getting one number, which means that that's the diameter of the shape. And because it's one number, your assumption is that this shape is spherical. And not only that, it's a spherical shape that doesn't change. It's not squishy, it's not soft, it's hard. And so one of the things that happened as I was working with the data is that I kept having 2 peaks. So I wasn't really getting one consistent number in size. So if this is size and diameter, right, this is probably around like 16, and then we're going up to like 105. So we're seeing micelles that are small and micelles that are big. But more than that, the distribution that we were looking for is showing some of them in intensity down here, and then some of them intensity over here. And this was really confusing to me at first because I thought that they would be kind of consistently one size, very similar to this orange peak right here, where we have a generalized mean, and then we kind of have like a standard bell curve. And I kept redoing the experiment. I did it 138 times to try and figure out what is going on with these peaks. And then I started analyzing the volume. And so what that is different than intensity. Intensity is what is reported in the literature. Volume is kind of how much of each of these peaks is taken up within that solution. And one of the things I noticed is that some of the volume of the shape in the smaller range, it actually took up more of the solution than the bigger range ones took up less of the solution. And so as I was reading, I found out that sometimes you can create micelles, which are those spherical shapes that are kind of like spaghetti, but sometimes you can create what's called worms. And I'll show you a picture of that in a minute. So in another paper, they did cryochem images on their work. And basically what they found out is they can have these little spherical balls, but they can also have these longer worm-like shapes. And you can kind of see them here that happen in self-assembly. And so my hypothesis kind of after doing this work is that we had both in our solution. So when we went to try and create micelles, we had both spherical shapes and worm-like shapes. And because I was only getting one number from the DLS machine, it could be that the worm was turned on its side sometimes, and then sometimes turned this way or turned this way or turned this way. And I think that's really important. And my understanding of this work, but also in working with students, is that often as a teacher, I draw things in 2D to try and represent for students a 3D world, and they don't totally get it. And I wasn't totally getting it. I was over and over again redrawing and going, what's wrong? Let me do another trial. What's wrong? Well, let me do another trial. And then when I started thinking in a 3D world, I realized, oh, this could be possible. And I love this idea that the data doesn't lie. So we have to keep going back to the data. So the kind of next steps for this project, you'll notice that I haven't talked about water dynamics in this one yet. We did put that spin label in, and so that's the next step on this project, is to start working on the water dynamics around this longer peptoid. For me, what I'm taking from this research experience is, one, just for me as a scientist, I was able to stretch and grow into this micro world when I've been living in a very big macro world. And so it really helped my brain understand my macro world at a greater level, and specifically in self-assembly of all living things. The other thing that happened was being in a lab rather than in a field work, which is what I've predominantly done and also mostly done with students, is that I noticed that Audra's group has lab experts. So everybody's working on a different research project, but there are people that know the technology in different areas better than others, and they'll ask each other questions about it. And I really want to take that back to my school, where students can become experts in certain areas, and they can kind of get certificates in certain areas to help out other students, specifically because we're pre-K through 8th grade. So it's a great learning and leadership experience. I also know that middle schoolers love self-assembling. They love choosing their own groups. And sometimes as a teacher, that's really hard because you want certain students to work with other students. So I've been brainstorming ways where maybe with this expert idea that they could self-assemble. Like, you need someone in your group that knows the microscopes really well. You know, someone in your group that knows how to like work with glassware really well, right? And so kind of self-assembly and trying things like that. I also loved that in the Siegelman Group's lab, that there were manuals to every instrument. And I realized for middle schoolers, those manuals probably need to be video manuals with a link where they could, if they forget how to use something from one lab to another, we have both a science lab and an engineering lab, they could look at that link and watch the video, and I could point them towards that to kind of help them be on their own. The other thing I learned that I will take back to my students is to start with controls. So when I was doing my multiple trials, I kept getting this large peak that actually wasn't a polymer peak. And it was from either dust or also mildew. The water I was using had some kind of impurity in it. And as I was doing it, I didn't know if that was my product or if it was something else. And I always have students build controls into their experiments, but I don't make them start with it. And I feel like if I would have started with it, it would have helped me go a little bit quicker on my multiple trials. I love also having students redo experiments with changes. I think for me in my curriculum, I've always had them mostly do an experiment and then kind of move on to the next one. But coming back the next day and doing the experiment again and modifying it really helped me to learn a lot more and have a lot more depth and understanding. I also like the idea of bringing all the class trials together and looking at the data, maybe on a Google sheet, and graphing it to look at those trials for trends. Something that I just picked up on this year because I was having a hard time visualizing was working in 2D and working in 3D. And I think I should bring that back to my school as well. It's like, how as teachers can we help students to model things in 3D, or how can we model things for them in 3D so they better understand, let's say, the water cycle or chemistry or all of these things. And I think it was a really good experience for me to do it that way. And then the last thing that I've talked to one of our elective teachers about, who's actually a chemist, is creating a new elective class called Materials Science. We often have students go through curriculum that we love, but we don't have them always create new things besides in our makerspace, which is with like paper and cardboard. And so I've thought about, like, what about if they're creating new materials? So they start out with like a slime, a chocolate, a soap recipe, a bath bombs recipe, and then they have to modify it and come up with an engineering plan on ways to make this better or more interesting or do something novel. And I think it would be a really interesting course for them. So overall, this was just an amazing experience where I felt like I was drinking out of a fire hose every day and learning so much. My mind was stretched and grown in lots of ways, and I'm just super thankful for this experience. Yay, any questions?